Today we're going to be looking at nucleic acids and how they affect the body. This is kind of an intro into talking about DNA, as it allows us to learn some vocab when I start talking about how the cell creates proteins and eventually is able to survive based on a single strand of DNA. Let's take a look at it. What are nucleic acids? Every cell needs a set of instructions for survival. The code for this survival is nucleic acid. Nucleic acids act as the information carriers for the cell. Together, they are able to make sure that the cell performs all the necessary functions in order to survive. What are nucleic acids made of? Nucleotides. These are the monomers of nucleic acids, contain one nucleobase which acts as a piece of a code. Nucleotides consist of 3 parts and are very small. However, when they're stacked on top of each other, they can create large nucleic acids such as DNA and RNA. What are the types of nucleic acids? A. DNA, deoxyribonucleic acid. Found in all cells is the blueprint for everything in a cell. DNA is found in every living organism, and between every living organism, DNA is different. Cells contain massive amounts of DNA as they hold massive amounts of information to ensure that every cell is able to survive. Is double-stranded. The 2 strands run opposite to each other. The 2 strands of DNA run opposite to each other with their matching base pairs. This allows to make sure that there is a maximum amount of information carried. Contains 4 bases: adenine, which we represent with an A, guanine, which we represent with a G, cytosine, which we represent with a C, and thymine, which we represent with a T. These are the 4 base pairs. You notice that they are very different in sizes. RNA, ribonucleic acid, found in all cells, acts to help carry out the cell duties based on the DNA. RNA acts to be the messenger for DNA. As you can see here, there's a molecule reading off a strand of DNA and creating a long yellow strand of RNA, which will be sent elsewhere in the cell. RNA's main role is to dictate to the cell what it is specifically that the DNA is instructing for the cell's survival. Is single-stranded. Only one side of the DNA is read at a time. RNA is different from DNA in the sense of one key thing is its structure. RNA is single-stranded, as you can see by this picture, whereas DNA is double-stranded. Contains 4 bases: adenine, which we represent again with an A, guanine, which we represent with a G, cytosine, which we represent with a C, and uracil, which represents a U. These are pictorial representations of the 4 base pairs of RNA. Notice there's no thymine, instead a uracil is there. Strawberry Extraction Lab. The goal of this lab is to observe what DNA looks like. Procedure. At your table, make sure you have the following: a graduated cylinder, cheesecloth, funnel, and glass rod. Come up to the front with your graduated cylinder, grab 1 strawberry, a plastic bag, and 10 milliliters of a buffer solution and test tube. At your desk, place the strawberry in a bag and mush for 2 minutes. Add the 10 milliliters of buffer to the strawberry mash. Knead strawberry again for 1 minute. Assemble cheesecloth, funnel, and test tube as funnels. Slowly pour the strawberry mixture into the apparatus. Come to the front for cold ethanol. Slowly pour the ethanol into the test tube. That again. The next lab that you're going to see is how to extract strawberry DNA from a strawberry. This video is pulled from YouTube, so ignore the amounts that she talks about. However, the procedure will still follow the same.
You'll need 1 strawberry, 1 Ziploc bag, 2 small beakers, a plastic funnel, 1 square of cheesecloth, a tube rack, 1 50 milliliter plastic tube, 5 milliliters of alcohol, a pipette, 1 small tube with a cap or a lid, and 20 milliliters of extraction solution. The extraction solution is made by mixing 900 milliliters of water with 100 milliliters of shampoo and 2 teaspoons of salt. This will make enough for your whole class. First, place the strawberry into the Ziploc bag. Then pour 20 milliliters of the extraction solution into the bag. Close and seal the bag, removing as much air as possible. Use your hands to gently mash the strawberry in the extraction solution for 5 minutes. Place a funnel into the 50 milliliter tube. Next, place a piece of cheesecloth inside of the funnel. Open your Ziploc bag and pour the mixture of strawberry and extraction solution through the cloth and into the funnel. Gently squeeze the cheesecloth. Once all the liquid has passed through the cloth, throw the cloth away and remove the funnel. Add 5 milliliters of 70% isopropyl alcohol. Hold your tube to eye level and observe. The DNA precipitate looks like fine white strands of cotton fiber, almost like spider webs. Now you can use the pipette to remove the DNA from the tube, look at it, touch it, or place it into another tube to keep. Now you've successfully extracted the DNA from a strawberry.